we are going to study that is page 368, translation. But before translation, I want to ask about the genetic code. What are the genetic codes? Anyone who can tell me? What do you know about genetic code? Everybody can know this. People wrote there was what the parents from genetic code. What is this? Okay, from the online student who know what are genetic codes? Anyone who know? When three bases occur in the sequence, nitrogenous bases, when three nitrogenous bases occur in a sequence and code for one amino acid, this we call genetic code or codon, right? Genetic code or codon, right? When three nitrogenous bases come encode for one amino acid. This is called genetic code. Now these amino acid will combine and will form in turn what? These amino acid will combine and it will form in turn protein. So now how that protein occur? That we are going to discuss, right? So the first point which we are going to discuss, that is about translation, right? First point we have to discuss, that is about what? That is about translation. Mean that three nitrogenous bases come together and they code for what? They code for one amino acid, right? So when one amino acid and then another three will come, it will code for another amino acid, then another three, it will code for another amino acid, and the chain will just start. And this chain will later on form what? It will form protein. So how this chain is formed? The first point in this protein synthesis we are going to discuss, that is about translation. What is the meaning of translation in just in common English, not in scientific? Translation, yes. Like I told you, translate in Arabic. What does it mean? What does it mean by translation? Yes, simple, right? Like you have this is English, right? So you want to translate it. What do you say in Arabic? So there is translation. So the same we have translation here, right? What will be translated? In this condition, what will be translated? DNA will be translated into RNA, right? Because like this book we have, the entire book we have, it is a DNA. Let's suppose one chapter we need, so that one chapter needs to be translated, right? Because we need only this one chapter, right? The one which you see on the whiteboard, so this is what? This is the translation of the book, right? So the same we have DNA, and from the DNA, a specific part need to be translated. There. Okay. Translation, the decoding of messenger of an mRNA message into a protein. So now we have DNA. From the DNA, we have take a short template, right? In the form of RNA, a short sequence in the form of RNA. Okay, we take what? We take a short sequence in the form of RNA. Okay. So now this short sequence of RNA, which we call messenger RNA, what we call it? We call it messenger RNA. Now this messenger RNA, which have the information which it take from the DNA, this information need to be copied and need to form protein, right? So message into a protein, the decoding of an mRNA message into the proteins. So the codes, the genetic codes, which is present on what? Which is present on the messenger RNA. Now these genetic code, which is present on the messenger RNA, they are going to decode, right? When they decode, what will happen? The required amino acid will come and it will attach on it, right? The required amino acid will come and it will attach on it, and thus it will form what? Thus it will form a chain. Ribosome use the sequence of codons in messenger RNA to assemble amino acid into polypeptide chain. As I told you that now we have messenger RNA, and on messenger RNA we have genetic codes. And genetic codes are for 3 amino acids. It is formed from what? It is formed from 3 bases, nitrogenous bases, right? Now these codes are going to be decoded. So decode means that these codes will attract its own amino acid, right? On the other slide, you will see that how it will occur. Look to the diagram. This we have what? This we have a messenger RNA right here. This is entire we have a messenger RNA, right? Okay. Now, if you look on the messenger RNA, so on the messenger RNA we have genetic codes, right? 3 amino acid will code for what? 3 amino acid will code for one amino acid. Okay. Now let's suppose these are our code. Now this need to be decoded. If we need to decode it, so what we will do? AUG. A stand for what? Adenine. U stand for uracil. G stand for guanine. So now at the first point which you have AUG, keep in mind this, right? AUG. Now AUG is required for what protein? AUG is the genetic code of which amino acid? In the previous lesson we took AUG. Go to the previous page, you have on figure 13.6 AUG. This figure, what AUG code for? Come here. Come here. Show us. Come. No. Show us code AUG. What it's code for? For which amino acid it codes? Come on, come here, so you can easily find. Come here. Do you know? Ahmad, do you know? AUG.
They are nitrogen bases, and the C is the amino acid.
Sorry?
AUG is the nitrogenous basis, and the C is the amino acid.
AUG stained code for which amino acid? AUG. You have on the screen the diagram you can find it easily. Huh? No, it's not about the C. You have to find A, then you have to look for U, then you have to look for G. So mean AUG code for which protein? What is this? Methionine. I told you in the start that AUG. Look, we don't have anything here, right? So from here it is going to start. So the starting codon should always be methionine. The starting codon should always be only methionine, right? Only methionine. So AUG code for what? For methionine. Now UUC for what it codes? So you can check UUC code for UC alanine. So it's code for what? It's code for alanine. U here, then go to the second circle, U and C. So it's code for alanine, right? So you can find from here and it is the total number of amino acid we have here. Right? Total number of amino acid we have here 20. Besides this, we have starting codon and stopping codon, right? So 3 we have stopping codon, 1 we have starting codon. It's clear. So now UCC and now AAA. So AAA stands for what? Go back to the figure and you can check from there what AA stands for. A A and A or lysine. It stands for code for lysine, right? So in this way you can find. So you can find the entire protein, right? The entire polypeptide chain. So the one which is given here, this one. The first is methionine. The second one was what? UUC. UUC. Alanine. Right? The second one is alanine and the third one? Leucine. Leucine or lysine? Lysine, sorry, not leucine. Leucine is not, it's not leucine, it is isoleucine, right? But this one we have lysine. So it's clear for all, right? Okay. So in this where they are going to do what? In this way they are going to form the chain. Now the next function we have, the next part which will be played, that is about what? That is about the transfer RNA. Transfer RNA. Each transfer RNA molecules carry just one kind of amino acid. Integudan, three unpaired bases, which is complementary to one messenger RNA code. Right? Now the function of transfer RNA is what? Or tRNA is what? The function of tRNA, or we can say transfer RNA, is that it can carry only one amino acid and fix it on the polypeptide chain, right? It can carry only one amino acid and fix it on what? And fix it on the amino acid, on the polypeptide chain of amino acid. Each transfer RNA molecules carry just one kind of amino acid. So means for one amino acid, we will have different transfer RNA, right? Transfer RNA will be different for all the amino acids. How many we have? How many amino acids? 20, right? But in this example which we studied, that's 3, right? So here we need only 3 transfer RNAs. But for long chain, so let's suppose the protein chain is made from, let's suppose, 2000 amino acid. 2000 amino acid. So how many transfer RNA we need? 2000, right? Because one transfer RNA can carry only one amino acid. Integudan, 3 unpaired base, which is complementary to one messenger RNA codon. Now, if you look here, right? So one we have codon and another we have integudan. Codon we have studied, but integudan we didn't study, right? So integudan is opposite to the codon. The 3 unpaired bases, which is not paired, that is called what? That is called integudan, which is a complementary to one messenger RNA codon. Now, if you look to the diagram, right? This we have what? This point, if you look, this is what? This is amino acid, right? This is amino acid. This part, if you look, right? This part, right? This is transfer RNA, right? Which will take this amino acid and will carry it toward what? And will carry it toward the polypeptide chain, which is going to form, right? You don't have book, right? But in the book, it's very clear. The diagram is very clear in the book. The process of translation, okay, you have here. How the entire process will occur, right? How the entire process will occur in the, or in the case of the protein synthesis, right? You can see here, a ribosome attached to a messenger RNA molecules in a cytoplasm. Now, what is ribosome? Ribosome is an organelle which is present in the cytoplasm, right? It's an organelle which is present in the cytoplasm. A ribosome attached to a messenger RNA molecules in the cytoplasm. Where it is going to attach? It is going to attach in the to the messenger RNA in the cytoplasm. This is the step number first, right? First step we have that when the messenger RNA leaves the nucleus and it enters to the cytoplasm. So in the cytoplasm, what will attach to the messenger RNA? Ribosome will attach to the messenger RNA. As the ribosome reads each codon of messenger RNA, it direct transfer RNA to bring the specified amino acid into the ribosomes. So now, when the ribosome reads the codon, right, the codon which is already present there, right, on the messenger RNA. So it's a direct transfer RNA to bring the specified or specific amino acid to ribosomes. So let's suppose this here we have AUG. So for AUG, AUG is which codon? AUG is starting codon, methionine, right? So opposite for methionine should be what? Integudan for methionine should be. For A it should be U, for U it should be A, and for G it should be C. So the opposite codon should be UAC. You will what? For AUG it will be UAC, right? So that will be what? That is the opposite codon, right? Now next one we have UUC, right? So UUC AAG. AAG, lysine, phenylalanine, right? UUC will code for what? UUC will code for phenylalanine. UUC, yes, phenylalanine, right? And so on. Now, AAA code for what? AAA code for lysine, right? So these reading will be occurred by the ribosomes. The function of ribosome, right? The function of ribosome is that they are going to read the codon, right? And when it reads the codon, so it gives message to the transfer RNA to do what? To take the specific amino acid which is required for the codon and attach on what? And attach on the polypeptide chain which is going to form. Okay. On at a time, the ribosomes then attaches each amino acid to the growing chain and break the bonds between the transfer RNA and the amino acid. So the first function of ribosome is what? The first function of ribosome is to read the codon. This is the first function. The second function is to give message to the transfer RNA for the concerned amino acid, that which amino acid is required. The third function is to break the bond between the amino acid and the transfer RNA and leave the amino acid on the chain and release the transfer RNA. Clear? Okay. What is the fourth step? Okay. The transfer RNA move into a third bonding site. So now the transfer RNA will move where? The transfer RNA will now move into a third bonding site. Mean to the next step, right? The RNA will be ready now for the next step. From which it exists the ribosomes. And it will remove the ribosome. The ribosome move to the third codon where the transfer RNA bring it the amino acid specified by the third codon. So on the process will stop, right? It will start. Point number 6 we have. The polypeptide chain continue to grow until the ribosome reach to stop codon on the messenger RNA molecule. Give me the example of stopping codon. Ibrahim, can you hear me? Who can hear me from the online student?
Yes, Mister, I can hear you.
Give me the example of stop codon. We are saying that the polypeptide chain will continue to grow until a ribosome reach a stop codon. So give me the example of any stop codon, any one. Give it me as what we can say it's amino acid. It's nitrogenous basis sequence. What should be it? Any stopping codon. In the diagram you have, if you see. So the stopping codon we have UGA. Stopping codon we have UGA we have, UAA we have, and UAG we have, right? So the one which you have in the diagram, that is UGA. UGA is the stopping codon. So when till which time the process of the amino acid, the messenger RNA will translate into codon, and then the transfer RNA will give signal to the the ribosome will give signal to transfer RNA and it will bring amino acid. Till which time it will continue? So it will continue till what? Till the stopping codon reach UGA, UAA, or UAG. So any one of these three codon come, so then the polypeptide chain will terminate, mean it will stop, right? When the ribosome reaches stopping codon, it releases both the newly formed polypeptide and the RNA molecules, completed the process of translation. So at this point, a messenger RNA is completed, and it will complete the process of translation, right? Now after this, we have after this amount completing the poly. I think it's stopped here. After this, now the protein will form, right? Protein, you can see this chain which is formed, right? So this is a small chain, right? If you are going to wind it into this polypeptide chain, actually it is the newly formed, we can say, messenger RNA, or it is the newly formed protein. So later on, I don't know in which chapter you have, but we will continue it from here again, that how protein will form from this. I thought it is in this chapter, but it is not here. Okay, if you have any question till this place, so you can ask me. The diagram which you can see in the book, so it is explained very clear there, right? Because it's all on the one page, so you can get it easily.
Mr.
Yes.
I will rejoin.
Sorry.
I will log out and I will rejoin.
I cannot hear you clearly. Your voice is not clear. You can write in N-books then. Okay, now the last point of this lesson which we are going to discuss, that is about central dogma of molecular biology. You can say central dogma of what? Central dogma, what is the molecular base of heredity, right? The central dogma of molecular biology, yes. The topic which you have that is about the molecular base of heredity. The molecular base of heredity. Now, if you look here to the first line, this one, right? Information is transferred from DNA to RNA and to protein. So your entire topic can be explained from these three words, right? Not even your entire topic. These entire chapter, we have the 3-4 chapters related to the DNA, RNA. It can be explained from the central dogma. Why? Because central dogma explains the entire molecular biology, right? What is DNA? First, you have to study what is DNA, right? How DNA is formed, the synthesis of DNA, the compartment, the components of the DNA, the function of the DNA. Then you will come to RNA, right? From the DNA to the RNA. So when you are going from the DNA to the RNA, so first thing you have to discuss, that is about what? That is about comparison of DNA and RNA. Like what is the similarity and what are the differences between RNA and DNA? Right, and when you discuss RNA in a detail, so then after RNA we have protein, right? So RNA will be translated, or we can say transcription. That is not given here, but RNA should be transcribed to what? RNA should be further transcribed into the proteins. So this all information we have to study in this. The central dogma of molecular biology is that information is transferred from DNA to RNA into protein, right? So this is the central dogma, that the information is transferred from DNA to what? To RNA and to proteins. Central dogma means central theme, right? Central theme, what we are studying, right? What is the purpose? Exception include retrovirus, which is HIV, where information is moved from RNA to DNA. Those they give you example of only HIV virus, right? So those organisms which are RNA organism, only we have in the case of virus, right? Like we have COVID virus. So COVID virus also contains. It also contains RNA virus, right? So in the case of RNA, it has exception. right? That then the information are going to transcribe or translate it from RNA to the DNA. Rest, for all other organisms, it should be from DNA to RNA and then to protein simply, and protein will perform the function. Now, here the point which we are going to discuss, gene expression. Gene expression is what? The way in which DNA, RNA and protein are involved in putting genetic information into action in a living cell, right? The functions which are occurring, right? The function which occur, like we are talking, we are walking, we are eating, our digestive system is working, our respiratory system is working, immune system is working. So all these are based on what? Or these are based on the gene expression. Like for example, if you look to the DNA, in the DNA we have what? In the DNA we have codes. And the translation of these codes into RNA and into protein, right? And then protein into different functions. These all are present, right, on the genes. So when it is going to translate from the gene, this process we call what? This process we call gene expression. Clear? Okay, now in the diagram which you can see down, right, they have, you can visualize it. DNA is present where? You can see this is DNA which is present in the nucleus, right? Now from the DNA, a short sequence of, this is the entire lesson which we study, right? The entire lesson which we study. So this is DNA, from the DNA, a short template will come, will unwind with the help of DNA, RNA polymerase, and after that, what will happen after that? DNA polymerase will form a small messenger RNA, and that small messenger RNA will travel out of the nucleus, right? Out of the nucleus to what? To the cytoplasm. So this is nucleus, this is the internal is nucleus. So when this transcription occurs, transcription means that the DNA, the short template of the DNA is converted into RNA, and this RNA, which is messenger RNA, it is going to transfer or go out from the nucleus, outside what? Outside the nucleus to the cytoplasm. So this area we have, this is the cytoplasm. Now inside the cytoplasm, translation will occur. Translation is which process? In which amino acid with the help of ribosomes will come and will attach on the codon, and it will form what? It will form a polypeptide chain of the amino acid. The first amino acid will always be methionine, and the last amino acid will always be the last amino acid will always be, it will not be amino acid, it will be stopping codon, right? It will be stopping codon. Starting codon we have methionine, but the last one we have, it is a stopping codon, and that is always what? That is always a stopping codon, which we have 3 different stopping codons. So any one of these 3 can come. Okay, the genetic codes. This is about the genetic codes we have taken just to look if there is any changed point. The genetic code is near universal for all, right? Because it's 3 genetic code we have the genetic codes which will only code for the 20 amino acids. So overall, it will be genetics. Universal, it will be near universal. Some organisms show slight variation in the amino acid assigned to particular codons, but the code is always read 3 bases at a time and in the same direction. So it is the same in all organisms, right? If you have any questions, you can ask me. Our session is almost finished, and the topic is also finished. Next lesson which we will start, there is about mutation. So if you have any point, any doubt, so you can ask me.